All right. I think everyone can hear me. So our next speaker is Dr. Ashley Zehnder, who, by the way, also studies aging in like unconventional models and, among other things, is also veterinarian. And she's the co-founder and CEO of Fauna Bio, a venture-backed biotech focused on rapidly translating insights from highly adapted species to create groundbreaking advances in human health. Fauna Bio was one of the first companies in the Longevity Found-backed Age1 accelerator and works with species with unique adaptation relating to aging and age-related diseases. Like I was mentioning earlier, she has a PhD in cancer biology, she's a veterinarian, and she has more than 20 clinic, oh, I'm just gonna let her speak, guys.
No worries. Thanks so much for the introduction, and thanks, Kristen, for putting on a great talk. So it's interesting. I'm sandwiched this morning between two amazing speakers talking about dogs and their models as aging. I'm going to channel Marianne, I think, from the panel yesterday and provide a little bit of a counterpoint to the argument about looking at animals as more rapid aging models. And instead, I'm going to take a moment to present the alternative, which is looking at species that have, through their natural evolution and through perhaps hundreds of millions of years of evolution, derived new ways to enhance their own longevity and survival, enhance tissue regeneration in a way that we can adapt for improving human health and human health span. So I'll start with what is perhaps maybe a bit of a tongue-in-cheek question for a summit like this, but when we're thinking about ancient evolutionary mechanisms of survival, it begs the question about what is longevity. And I would argue that at the end of the day, longevity is really the act of optimizing that survival, and optimizing that survival not only for an entire organism, but also for all the cells that make up those organisms. So really what I'll get around to as we go through this presentation is what are the different pathways and ways that different species have optimized to combat cellular regeneration and the damage that inevitably occurs as animals live sometimes amazingly long lives. So I'm going to take a moment to explore some species that have really optimized ways to enhance their own cellular survival. One group of those species are the hibernating mammals, and this is really where we focus a lot of our attention at Fauna Bio. So what's really interesting, I am a veterinarian, my background is in exotic species, I'm a boarded avian vet, but until I started this company with my co-founders, I did not appreciate the diverse and varied biology that is hibernation and how amazing it is in terms of a phenotype. So hibernation, actually, if you look back through the fossil record, even our earliest mammalian and pre-mammalian ancestors show evidence of being able to use torpor, which is really the periodic lowering of metabolic rate to survive in periods of resource scarcity. And this is something that, again, very ancient mammals have used to survive, perhaps even events like the asteroids that killed the dinosaurs. But what this suggests is that these are genes and pathways that are common to all mammals, including primates. There are animals like this fat-tailed dwarf lemur up here. I don't know if I have a pointer. There you go, up here, this guy up here, that can hibernate very similar to species that we're more used to thinking about, like the 13-line ground squirrel. This is important because what we do at Fauna is look at genes and genetic networks that are activated at time points where these animals are protected. It turns out these are the same genes and pathways you and I have that they use in slightly different ways. And when you look across hibernators as a group, accounting for obviously their lifespan differences and their life cycles and life strategies, generally around a 30 to 50% lifespan advantage for species that hibernate compared to similarly sized mammals that don't. And people have looked at why this may be the case. And so there are studies that are starting to come out really more since the mid-2000s and even as recently as this year, looking at what are the mechanisms that this group of species use to protect themselves and enhance their own longevity. So it really breaks down into a couple of key pathways. Ways to protect the genome and improve genome stability, but also ways to protect cells from damage. And this is something I also didn't appreciate until working on this company, is that hibernation itself is actually quite damaging. And we'll talk a little bit about why that is the case. Some of what we see even in our own data is looking at genes that are going up in species during time points when they're hibernating. So these are genes that are going up in heart RNA-seq during hibernation time points, we see that these are enriched for genes relating to response to DNA damage, mitochondrial stress, DNA checkpoint repair, all DNA repair type mechanisms that are enhanced during the period of hibernation. This correlates quite nicely to studies that other investigators have done looking just broadly at lifespan across a range of species. This is a study from the Gerbernova lab that was published just in June of this past year, looking at 26 different species over a range of lifespans, mostly in the rodent clade, and the highest enriched set of genetic pathways here were related to DNA repair. There have also been studies looking at ways, at a very basic way of animals elongating their telomeres to help protect themselves from damage and respond also to some telomere shortening that's observed during species during periods of oxidative stress and hibernation. So across multiple different species during and one hibernation cycle itself, increasing in a relative telomere length over a hibernation cycle, but then also over a lifespan of an animal as they pass sexual maturity, being able to increase the length of their telomeres. I am not an expert in epigenetic clocks, and I will not pretend to be, but there have been a couple of studies looking at epigenetic aging specifically during the hibernation period, one in marmots and one looking at big brown bats, showing that over the hibernation time points and over a single period or over lifespan, we get a slowing of this epigenetic aging at marks that have been defined for these species. But what I think is actually really much more interesting for our purposes, because it relates directly to human damage that we see during diseases, is looking specifically at how these species are able to regenerate at a cellular level. So this is a study that was published in 2015, and this is data that we see ourselves in our own data from 13.9 ground squirrels, looking at genes that are overexpressed as animals go through the spring time points into the fall transition, and then through torpor and these interbout arousals, which I'll talk about in a moment, that are part of that damaging phase of hibernation. And what we see is that these animals are able to really reactivate some of these fetal genetic programs that actually are quite similar to what James was mentioning yesterday when we talked about Cambrian brio, that these animals as adults are able to leverage and utilize neonatal regenerative programs that can make their hearts and other organs be more likely to repair damage. And we see that ourselves. So I mentioned, these are genes that are going down here in green at time points where we can see physiologically that hibernators are protected from damage. These then, we compare this directly to networks from neonatal mice at time points where they are still able to regenerate at the very early neonatal time points. And so we can see a really terrific enrichment here of adult hibernators and also neonatal mice, again, that retain this regenerative capacity. So I think then the question really becomes, there's a lot of really interesting insights we can get from these types of species, but that is a biological question. What we're really interested in at Fauna is how do we translate this really extreme and highly optimized biology into benefits to help humans live longer and healthier. And our answer to that is really our platform called Convergence, which integrates all the genomic data, links it to human diseases, and helps us optimize therapies based on this evolutionary understanding. So Convergence has several different components to it, starting with ORCA, which identifies these specific disease resistance signatures in the RNA-seq signatures from different tissues and different time points. CETUS then generates gene scores across different data sets looking at conservation. How highly conserved are these genes across multiple hundreds of species, including humans, with the idea that if nature has conserved a pathway across hundreds of millions of years, it's much more likely to be functional. We validate all of the findings that we have in primary human cells and cell lines and also in animal models of disease, and we integrate a significant amount of human genomics data in the work that we do. So we've integrated data from the UK Biobank and other sources, and altogether about 40% of the data on this platform is actually human. Centaur is our knowledge graph that then integrates all of the multi-omics data that we collect, so whole genomics, transcriptomics, proteomics, epigenetics, and integrates that with external data sets to be able to more rapidly predict the links between these animal protective genes and human diseases. And then we have a module called LEO that allows us to map genetic signatures that we find in these protected species directly to small molecule compounds that we can optimize for our own drug programs. So I'll take a minute to walk you through what is really a very remarkable piece of physiology. For those that have never seen this before, this is what a body temperature trace of a 13-line ground squirrel looks like during hibernation. It was actually this temperature trace that convinced our CSO to drop the study of human genetics and say, honestly, I'm going to study hibernation because it's way more interesting, right? One graph can change your life, right? So, these animals start out like any other respectable mammal at around 37 degrees Celsius. But then in the fall, they drop down to almost freezing, so they'll stay around 4 degrees Celsius. When they're in this period of deep torpor, their metabolism is about 5% of normal, their heart rate is 5 beats per minute, their whole body is operating about 10% the normal oxygen level. Their neurons physically retract and they become functionally brain dead. They become hyper-tau phosphorylated. They are little frozen suspended balls of fur, right? And we've held them. We took a field trip up to the University of Wisconsin in Oshkosh, where there is the only research colony for these ground squirrels. And it's really amazing to hold these animals in torpor, and you feel like you're holding an ice block, but it's a living mammal. But they can only do it for a couple weeks at a time. And this is really the key difference between these small-bodied, deep hibernators and things like bears, right? Bears do it slightly differently. We're talking about ground squirrels. So these animals can only do this for a couple of weeks at a time, and then they rapidly rewarm over the course of just about an hour back up to 37. And so their metabolism increases 235-fold in that 1 hour time span. My CSO likens it to going from a dead sleep to biking the Tour de France in an hour, right? And you do it every 2 weeks. And you can imagine this is actually quite damaging. So you get this rush of warm oxygenated blood into every organ in your body. Brain, heart, liver, kidney gets this rush of oxygenated blood. That can actually be quite damaging, right? It's very similar to what happens if you have a heart attack. You go to the doctor and get your heart attack unblocked. You get a rush of warm oxygenated blood back into those parts of your heart that were previously unperfused. That's called reperfusion injury, and it's actually quite damaging. So these animals survive the equivalent of 20 to 25 reperfusion injuries in the course of a single hibernation cycle. I don't know about you guys. I don't know anybody that survived 20 to 25 heart attacks in a single year, right? But they do this every year. What is so cool is that they're not made of steel. They actually do take on damage. So this is, apologies for those who are not used to looking at histology. This is a cardiac cross-section of hearts during different phases of hibernation. So during early summer, during the fall, and in winter, they actually look pretty normal. The muscle fibers are really organized. There's not inflammation. But during these peak time points, during this arousal, you start to get evidence of damage. You get wavy fibers, you get increase in collagen deposition, you get damage that if a human cardiologist looked at it would say these are irreversible signs of damage. But they reverse them every hibernation cycle. So what we do at Fauna, let's see if this will play. Come on, you can do it. This is supposed to play. What this is supposed to be.
I think it's the pointer. If you switch back to the cursor, you should be able to start it. But...
Wait, okay. Oh, okay, hang on. How do I change it to the pointer? Wait. Oh, no! It worked on the thing. It's not going to play. There you go. No, it's going.
You got it?
Nope, it's perfect. Okay. All right. So this is just a zoom through in part of our platform called Orca, where we use this to directly compare these protective signatures at time points where we can see physiologically the hibernators are protected from damage. But then really critically, we can overlap that with the same genes from humans with diseases. So here we can overlap it with, and blue are the hibernator genes that are down at time points where they're protected. In the human heart failure patients, these are the rings that here are in yellow. And so that's where, let me get my pointer back. Can I get my pointer back? There we go. And we can use this then to enrich for what are the types of genetic networks that are oppositely regulated in these species where we see this really profound protection from damage. Now I gotta figure out how to advance my slides again. Oh, come on. There we go, now we're good. All right. But this is all somewhat theoretical until you actually test these things on the bench, right? So what is amazing about what we've been able to do with this system is that we can then take genes that we identify with this opposite regulation approach. We can knock those same genes out of human cardiac cells where we put them in low oxygen and we deprive them of glucose, and we can double their survival in vitro. I'll get my pointer back. We can also then take those same genes, try to get my pointer back. There we go. Okay. Pointer's back. We can take those same genes and knock them down in species where we physically perform a coronary ligation. So we can simulate a heart attack in a species like a rat. You can see here in the control we have a very large scar and a heart that is not going to function properly after that. But here we can knock these genes down and we can almost fully protect these hearts from damage. This is not just a cardiac phenomenon, so I don't want to give the impression that Fauna Bio is a cardiac company, because actually our lead molecule right now is in pulmonary fibrosis, and I'll talk about that in a moment. But we can also look at different parts of the brain. So this is single nucleus RNA-seq that we did in hypothalamus from hibernators at time points where, again, their brains are going into this really suspended animation state, and we identified those cells that are able to stay functioning even when these animals are almost frozen and lacking of oxygen. So it's a little bit like thinking about your computer's motherboard, right? When you close the lid, parts of your computer motherboard have to stay a little bit awake so that when you open the lid, your computer can come back on. So parts of that brain still has to function, even in a cold and lack of oxygen. So we look at what are the genes that are correlated with those neurons that are going to torpor but have to stay functional. And so we found a gene called 264G that is highly conserved across practically every species that has the same hibernation patterns. Very enriched conserved gene, highly conserved in people as well. And we then, what was super cool, is that we can overexpress, these are just control cells at normal 37, they're all happy, that's all good. But we can take this gene and we can overexpress it in human neurons, we can put them in the cold, and we can deprive them of glucose and lactate. We can force them to switch to using beta-hydroxybutyrate, which is the preferred fuel substrate of hibernators during torpor, and we can make them happy at 4 degrees Celsius. So this, again, biology that ports really nicely across into humans. What we then do is to try to say, okay, how do we actually get small molecule therapeutics that can mimic this protective signature and we can use to jumpstart our own preclinical pipelines? So here we've defined these expression signatures that are protective in hibernators. We use a module called LEO to map them to a database of drugs that have been used to treat many different human cells. And then we find small molecules that can essentially mimic the natural protective signatures of these hibernators. And this is just one really kind of quick snapshot of a molecule that we're working with now called FON1003, which is a new mechanism for pulmonary fibrosis, which seems to be active in cells that are responsible for sensing and responding to hypoxia, right? Which makes a lot of sense, highly conserved mechanism. But when we treat animals here, these are normal mice treated with bleomycin to induce a pulmonary fibrotic injury. We can treat them instead also with 1003, and we can really prevent a significant amount of damage in these animal models. And so based on this strategy of learning from natural animal protective mechanisms, we built a really diverse pipeline of therapies. Our lead, which I mentioned, is a lead optimization for pulmonary fibrosis. But we also have looked at building programs around cardiac failure and cardiac fibrosis, which is of interest to a couple of pharma partners that we're talking to. We are working right now, we disclosed a partnership with Novo Nordisk where we do target discovery around obesity. I mentioned these species can increase their metabolism 235-fold in an hour. There's no other model on the planet that can do that. And obesity companies are very interested in figuring ways to improve human energy expenditure and enhance obesity treatments. But we can also look at things like the GI tract, we could look at the brain, we're starting to look at kidney. And again, these are organ by organ protective mechanisms that we can look at to identify new novel therapies for regeneration. I will say that this is kind of a rich, untapped resource for discovery. Again, even as an exotic animal veterinarian, I did not understand how cool hibernation was. But all of the work that we've done to date has been primarily from the species, the 13-lined ground squirrel. But this is a process that actually we can replicate across other protected species. So we have collaborations already with a group at UNLV who works with a very ancient mammal called the tenrec, which again is able to, even better than the ground squirrel, regenerate heart cells much later in life. We're starting now to even work with non-hibernators that also have an evolutionary mechanism of protection. We have a collaboration with a group at University of Florida that looks with spiny mice who have ways to repair tissues in their body without fibrosis, including things like the brain and the spinal cord, which have evolved as ways to evade predation. So there's this whole new strategy of saying, again, let's not just think of animal models as animals that maybe age faster than we do and we can study that process more rapidly. We're actually sort of taking the counter argument which says, well, actually, let's look at species that through their natural biology either age slower than we do or resist the kind of damage that we see during human aging. Oops. There we go. And this is, again, sort of tip of the iceberg situation where there are many other species where you can take this approach. Oh, clicky. Now it's doing something funny. There we go. Oh, my God, it won't click. Thank you. Saved by the box. So I am one part of Fauna Bio. We've been really fortunate to attract and work with some of the best folks in the industry in terms of being able to go from early genomics through to therapeutics discovery. Brian Berkey joined us as our head of therapeutics this year, had a 20-year career at Novartis with Tom Hughes, again, using early stage genomics and putting drugs into the clinic and working on approved drug programs. And so it's been a really interesting mix of folks who are dedicated to working on vertebrate genomics, hibernation genomics, drug development, and really bringing all of these kinds of expertise together to really build a new and different kind of drug company. And then, of course, what do you do when you're Fauna Bio and you need to do a company offsite? You go to Safari West. So we all went out and had a great time studying all the different animals and getting to know each other better. So thank you so much. I guess I'll take questions before I run off the stage. Yeah.
Seems kind of obvious, but have you looked into cryonics in terms of like applications for the fusion work that you're doing?
That's a great question. It's not something we have looked at specifically, but we are talking to companies that are kind of working in that space because I think there are a lot of parallels to reperfusion injury and thinking about that. We've been looking sort of more near-term medical applications, but I think it's an interesting field to look at. Yeah.
So you've obviously
I see you. Yeah.
sacked this 13 lines of ground squirrel at various points in the cycle of cooling and
Yep.
warming up, right?
Yep.
For the transcriptomic insights that you got. Do you have tissues to do metabolomics? Because I suspect that you're hitting the NAD system in there somehow, and I'd love to look at that.
Yeah, we do have we have tissues from the original biobank, but as I mentioned, we work with the University of Wisconsin in Oshkosh, and so that is the one ground squirrel colony. We actually, another offsite we did was go out and practice ground squirrel trapping. They all live in cemeteries, by the way, which is really creepy. But yeah, so we have access to living tissues, and we're doing prospective collections for our own biobanking, but also through some of these pharma partnerships. Yep. Matt, you had a question.
Yeah, so super interesting. I love the unique approach that you guys are taking. The question I've got is, in that sort of cycle of rejuvenation or like the cardiac reprogramming that you showed, does that ability to rejuvenate or reprogram decline with age? In other words, as the animal's going through the cycles, as they get older, does that become less effective? And then, sorry, related to that, your genetic or small molecule leads, have you tested those in older mice for the ability to have these effects?
Both great questions. So the thing about the 13-line ground squirrels is that you don't get a lot of naturally old ones because everything eats them. So there are not a lot of natural aging studies in some of these species unless they're in the research colony, and then we tend to use them. So we haven't looked at the age relatedness of that. Like I said, these are all adult, usually at least second or third season animals that the biobank is from. Some from maybe 1 or 2 seasons in the research colony, but then they always bring in fresh caught. So we don't have this sort of lab artificialness to it. In terms of the mice, because I missed the second question.
Well, so I was just thinking, if that regenerative process declines with age,
Sure.
inability to regenerate, would the same factors work in an aged mouse or
Oh,
human?
that's a good question. Yeah, we'd have to look at the factors first, and then we'd have to test the question. But yeah, yeah.
Have you looked at all at sleep cycle in the mice or quality of sleep with this?
There's actually quite a bit of science looking at the links between kind of circadian rhythm and circannual rhythm, right? They're not the same, but they are somewhat related. So we do see enrichment for things like certain clock genes, and enrichment for some of these genes that are responsible for circadian rhythm. There's been some interesting papers coming out actually showing some circannual effects on diseases like Alzheimer's in humans, and I think people are sort of underappreciating the effects of our own and our circannual rhythm on some of those diseases. It's not something we've looked at super deeply, but there's interesting data to be looked at there for sure.
I think
Yeah.
we should stop.
One last one. Okay. Okay, last one. Okay.
So, coming back to human aging,
Sure.
we actually live a pretty long time.
Sure.
So, are there, this made me think, are there like hidden things that we don't know about us that are kind of analogous to the hibernation type things that we don't really understand yet?
Totally. I think that is totally true. And I think, like I said, the revelation for me was looking at those phylogenies and saying, look, these are the same pathways that we have. It's not that we're putting weird squirrel genes in people. We're not. Okay. We're just looking, it's really just a different way to look at our own human genomics through this lens of extreme evolution. And so, yes, these are all the same pathways we have. We just need to figure out how to turn them on at the right time. Yeah, I agree with you. Thank you so much.